right, so it's 3:10. We're going to get started. I want to say good afternoon to everyone and welcome to the Iowa Nutrient Research Center Spring Seminar Series. I am Kay Stefanik, Assistant Director of the Iowa Nutrient Research Center. This semester, our seminar series focus on phosphorus. We have 4 speakers lined up to present the research on various aspects of phosphorus movement, measurement, management, and reduction, as well as the impacts of phosphorus on water quality. The seminars will be held the 4th Wednesday of every month from 3:10 to 4 p.m., both in person and virtually. The first speaker in the series is Dr. Justin Chaffin, who I will introduce in a minute. The speakers for February is Dr. Barbara Cade Menon with the Swift Current Research and Development Center in Saskatchewan, Canada. The March speaker is Dr. Antonio Mallarino with Iowa State University Department of Agronomy and Iowa State University Extension and Outreach. And then our final seminar speaker presenting in April is Dr. Chad Penn with the USDA Agricultural Research Service and Purdue University. Before we get into the seminar, I just have a few housekeeping items. First, this seminar will be recorded and posted to the Iowa Nutrient Research Center's website, along with past seminar recordings. During the talk, if you have questions for Dr. Chaffin today, please type those into the chat box and I will relay them to Justin at the end of his talk. So to kick off our spring seminar series, we have Dr. Justin Chaffin. He is the research coordinator of Stone Laboratory and Ohio Sea Grant at The Ohio State University. Dr. Chaffin earned his PhD from the University of Toledo and has been with Stone Lab since 2012. His research focus is on Lake Erie phytoplankton ecology, specifically linking experiments and models to predict cyanobacterial bloom toxicity, determining drivers of benthic algal blooms, examining the effectiveness of data buoys at measuring cyanobacterial bloom biomass, rapid microcystin testing, and examining saxitoxin production in inland lakes. Dr. Chaffin's talk today will be on Lake Erie cyanobacterial blooms, overview of blooms and ongoing research. So with that, I will turn things over to you, Justin.
All right, thank you, Kay, and thanks everyone for joining in. I'm going to talk a lot about Lake Erie, but a lot of the things that we learn on Lake Erie can be applied to other lakes. It's some of the relationships that I'll talk about for Lake Erie are might be specific to Lake Erie, but the data that we use, you can look at similar methods and similar data for each individual lake to see if the similar trends we see for Lake Erie hold elsewhere. So I'm going to talk a lot more in general about Lake Erie and highlight a lot of recent research. And then I'm going to get into a little bit of my own research looking at bloom toxicity. So let's see if I can get the thing to advance. It's always a trick to find which button to push. There it goes. So most algae are not bad for lakes. And algae are tiny plant-like organisms that live in the water. Algae are responsible for 50% of the Earth's oxygen. So every other breath you take, you should thank algae. And most of that oxygen comes from the oceans. Algae are the base of the food web. Good algae are food for zooplankton, and zooplankton are small shrimp-like creatures. And then the zooplankton are consumed by small fish, and then small fish are then consumed by larger fish. So algae are critically important for lakes, and there are hundreds or thousands of species of algae in lakes, rivers, and the ocean. They come in all different sizes, shapes, and colors, but they all need the same thing. They all need sunlight. They need fertilizers like carbon, nitrogen, and oxygen. Phosphorus and nitrogen and carbon dioxide. Lake Erie produces most fish of the 5 Great Lakes because it has the most algae. Lake Erie has the warmest waters during summer, and it has the highest nutrient concentrations. Those two factors interact to grow a lot of algae and support a highly productive fishery. A general rule we say at Stone Lab and Ohio Sea Grant is that Lake Erie has 50% of the fish but only 2% of the water. And on the opposite extreme is Lake Superior, which has 2% of the fish and 50% of the water. However, too much of the wrong type of algae is harmful. And I define harmful algal bloom based on these, now what I have here. The word harmful means it has the potential to produce a toxin. It may not be producing a toxin right now, but it has the potential to. Harmful algal blooms have harmful impacts on the ecosystem, and they have harmful impacts on human health and the economy. The word algal just describes what type of organism we're dealing with. In freshwater, we're dealing with cyanobacteria, which are also called blue-green algae, but marine systems have red tides, for example, which are a harmful algal bloom. And the term bloom is not a scientific term. It's a term that is used to describe a biomass that exceeds what's normal. So lakes shouldn't be green. The oceans shouldn't be red. So harmful algal bloom is a biomass of algae that has the potential to produce toxins and have negative impacts on human health and the economy. Harmful algal blooms are a global problem. It's just not a US problem or Ohio problem. No matter where you are, blooms are due to excess nutrients. However, how those excess nutrients get into the lake may differ from lake to lake or river to river. Basically, if something is put on a field or lawn to grow plants is used in general, crops or grass or trees, whatever you want there, it will grow algae once it gets in the water. So fertilizers, manure, lawn care, untreated sewage, all those will grow algae once they get to the lake. Some problems associated with blooms. They produce toxins, which I'll talk about in a little bit more depth later. Can be taste and smell problems. The water might have your drinking water might have an earthy flavor or a smell to it. Aside from if there's toxins in it or not, you can just smell the water. It smells a little funky. But also, if you're walking next to a lake that has a bloom, you'll get a whiff of a sulfur smell. Negative economic impacts. Lakefront restaurants and shops don't want green water in front. We work closely with charter captains in Lake Erie. There's over 1,000 charter boat captains in Lake Erie, and they report to us about a third of their business is lost when Lake Erie has a bad bloom. Cedar Point, which is a large amusement park in Sandusky, Ohio, sitting on Lake Erie, complains about blooms in Sandusky Bay. In 2014, August 2014, Toledo, Ohio was placed under a do not drink advisory because there was microcystins in the tap water that exceeded a safe level. And it was estimated that that 3-day do not drink ban cost at least $65 million. Blooms decrease property value throughout the United States. An estimate that's over 10 years old now is that blooms cost us $2.2 billion per year in lost property value. There's also negative food web impacts. Cyanobacteria are not consumed by the zooplankton. And during really thick blooms, the good algae dies off. So the bottom of the food web can really be starved. This image here is an image of a bloom in China 2 weeks before the 2008 Summer Olympics, and I think it was in Beijing. This is the pond where they did the rowing or the crew in that sport where they row the boats. They had all the village children grabbing a filamentous green algae out of the water and putting in these sacks to try to clean up. So blooms are truly a global problem. We talk a lot about harmful algal bloom toxins. One of the most toxic algal bloom toxin we have is microcystin. This list of toxins here, the green ones are cyanobacteria toxins. And then all four of these cyanobacteria toxins are more toxic than DDT or cyanide or atrazine. So these toxic compounds the blooms produce really are toxic. And when microcystin was first discovered in the early 1900s, it was termed fast death factor. They knew if they injected microcystis cells, which is a type of blue-green algae, if they injected those cells into mice, the mice died really fast. So they named it fast death factor, then later identified the compound and called it microcystin. So Lake Erie, if you're not familiar, in Northern Ohio, it borders US and Canada. Windsor's here, Detroit, City of Toledo, Cleveland, Pennsylvania, and Michigan are further off to the east. The Lake Erie blooms occur during summer. They can be seen by satellite image, and the blooms occur in this western corner of the lake, which we call the Western Basin. So we divide Lake Erie into three basins: the Western Basin, which is shallow, this large central basin, and then a deep Eastern Basin, which isn't on this image. So the Western Basin near Toledo, where the lake has blooms. A brief history of Lake Erie harmful algal blooms. In the mid-1900s, Lake Erie was declared a dead lake by Time magazine. There was bad blooms, there was fish kills, and the Cuyahoga River caught on fire several times. That prompted the U.S. EPA to form the Clean Water Act and the Great Lakes Water Quality Agreement. After those regulations, the lake recovered. Lake Erie became the poster child for ecosystem recovery. It became the walleye capital of the world, and there were no blooms in the 1980s. Since 2002, annual blooms have been documented. 2011 was the worst bloom in history at that time. And this is the background slide, or the background image of the slide shows a small boat going through a thick scum. This cyanobacteria can float on the surface, form these thick scums on calm days, and then when a boat goes through it, it creates a green wake. 2014 was the Toledo water crisis, when Toledo was told, do not drink your water. 2015 was the new worst bloom in history. And 2018, the Ohio EPA declared Lake Erie impaired due to HABs. And the word impaired, I have it with a capital I here, that has some legal implications. I'm not going to go into that, but it just means that the state is more serious about fixing up Lake Erie. So why do these blooms occur in Lake Erie? If you look at the 5 Great Lakes in cross section, we see that Lake Erie is by far the shallowest of all the Great Lakes. And the Western Basin is the shallowest basin of Lake Erie. The average depth of the Western Basin is only about 24 feet. The other 4 Great Lakes have maximum depths over 700 feet. Lake Superior maximum depth is 1,300 feet. Another reason why Lake Erie, if you look at the watershed and the land use within that watershed of the Great Lakes, we see that Lake Erie has the most amount of urban, so the most amount of people, has the highest amount of agriculture, and it has the least amount of forest. And only about 10% of the wetlands remain in the Lake Erie watershed. Wetlands are critically important because they act like a kidney to strip nutrients out of water before it gets to the lake. Only 10% of the wetlands remain in Lake Erie. Again, if you back out, look at that same data in a map, there's lots of orange and yellow around Lake Erie and red, which is urban. Also to note that the Maumee River watershed, which drains into the Western Basin, is the largest watershed within the Great Lakes Basin. And it's over 80% agriculture. You have the largest watershed draining into the smallest basin of the Great Lakes. So land use highly impacts small basins like Lake Erie, like the Western Basin. So we talk a lot about phosphorus, but there's different forms of phosphorus that we talk about. One is total phosphorus, and then a second common one is dissolved phosphorus. So if you were to go out to a stream, lake, or a river, and you scoop up a water sample, and you measure phosphorus on this water sample with all the sediments and algae in it, that's what we call the total phosphorus. If you run that sample through a filter to remove the sediments and algae, you end up with a clear sample. And if you measure phosphorus on this filtered sample, is what we call dissolved phosphorus. And the dissolved phosphorus is what is critically important, because 100% of this phosphorus is available to support algae growth right now. Phosphorus that's in its particulate form or is trapped on the filter is extremely hard and extremely slow process before it can be used by algae. So total phosphorus is important, but also the form of phosphorus, and especially dissolved phosphorus. So if we look at dissolved phosphorus concentrations in the Maumee River over time, we see a downward trend through the 70s and into the late 1990s, and then an upward trend. If we look at when Lake Erie had blooms, we had blooms in the 70s. We had no blooms in the 80s and early 90s, and we're back to having blooms now. So the Maumee River phosphorus concentrations have a huge impact on Lake Erie blooms. This downward trend was due to improved sewage treatment and less phosphorus fertilizers being used by farmers. The upward trend here is a result of changing in fertilizer application and tilling practices and tillage, but not more phosphorus being applied. So a larger percentage of the total phosphorus is shifted to a dissolved form. And I'm not a soil scientist. I'm not going to get into why this shift has occurred. Hopefully some of the other speakers will talk about dissolved phosphorus and soil use. Within Lake Erie, there are other types of cyanobacteria. I mentioned the Western Basin, where all the focus has been is microcystis. That's the genus that occurs in the Western Basin. Sandusky Bay has Planktothrix, which is a very shallow basin. It's highly mixed. Sandusky Bay acts more like a inland lake with a lot of Planktothrix. And occasionally the Central Basin will have a low density but highly spread out Dolichospermum bloom. But for today's talk, I'm only going to talk about microcystis in the Western Basin. And the Western Basin also has a benthic cyanobacteria. Benthic meaning it grows on the bottom, called Lyngbia. And this Lyngbia likes wave protected areas. It likes nutrient rich wave protected areas, and then it can wash up on shore in extremely high masses. So these blooms are visible and quantifiable by satellite. This is a MODIS image here. You see the true color. This is in late October after the bloom has spilled out of the Western Basin. But researchers at NOAA have figured out how to take a true color image like this and convert it into data. And in fact, this is available for almost any lake that is larger than a couple square acres, I believe. I don't know the exact size, but if it's bigger than a football field, you might be able to see it from satellite. So that satellite has been in use since 2002, so we can go back in time and see when Lake Erie had blooms. So in 2002, there was not much of a bloom. 2003, fairly large bloom. This is the bloom that got the scientists' attention, especially near Toledo. We've seen this large microcystis bloom kind of pop up almost out of nowhere. Then you have a couple of years of low blooms. Then you have a relatively large bloom, another large bloom. And you can see some year-to-year differences. 2011 was a pretty bad bloom. 2012 was a small bloom. Again, then 2016 was a small bloom. 2015 was that worst bloom on record. So we have this satellite data that goes back in time. And if you take this satellite images and you look at it every day and add up all the pixels, you can calculate a biomass index. And that's what's shown on this graph here. So like I said, 2003 was a fairly large bloom at that time, but 2011, 2015 were much larger blooms. And you see there's year-to-year differences. And we also do which I'll talk a little bit about later, but we also do a harmful algal bloom forecast where we can now forecast the bloom biomass. Looking at this data, this also shows you Lake Erie can recover in 1 year, but then it can go right back to being a large bloom the following year. So there's really no year-to-year carryover. So a bad bloom this year will not result in a bad bloom next year. So if you take that chart, turn it into a scatter plot, and then on the x-axis you plot the total phosphorus load from the Maumee River March through July. So not early spring, not the whole year, March through July. So when you have that spring thaw or the spring rains and into early summer, how much rain we get drives how much phosphorus is lost from the watershed. And then that total phosphorus load is correlated with the overall bloom size. 2015, or 2012 shouldn't be here. 2013, 2015 were very wet springs. 2012 was a drought. So the amount of phosphorus we get March through July explains the size of the bloom. We can also look at this and set a phosphorus loading target, which the new Great Lakes Water Quality Agreement in 2012 did. We want to be to the left of this line. So the new phosphorus loading target is 860 metric tons, which also equates to a 40% phosphorus load reduction. That 40% is used more often in media, but that 40% corresponds to if you just take the average load from all these years, 40% of that is 860 metric tons. That's what I just said. So what might a 40% phosphorus load reduction do? Very similar chart where the gray is the observed. And if we reduce phosphorus by 40%, the new bloom size would have been what we see here in green. There's going to be some years where we still exceed the target bloom, but the goal is to not exceed this bar here of 3, not exceed it 9 out of 10 years. So take the 2008 bloom and make it look like the 2012 bloom. The larger 2011 bloom will then look like the 2008 bloom. So again, if we meet that phosphorus loading reduction target, we'll only have a bloom of this size 1 out of every 10 years. And that's just due to rainfall that we get. Some years we get a lot more rain than others. We also know that majority of the phosphorus enters Lake Erie during storms, during and after storms. So the chart on the left, cumulative discharge from the Maumee River. So basically every time there's a spike on this graph, it indicates a rainstorm. And if you look at the total phosphorus load, every time there's a spike in rain, there's a corresponding spike in phosphorus loads. So this is how we make that phosphorus or that harmful algal bloom forecast. We track the amount of phosphorus that's coming down the Maumee River daily sometimes 3 times a day during storm events. And then once we know how much phosphorus is entered into Lake Erie in early July, we can use precipitation models to forecast what it might be, and then use that relationship between phosphorus and bloom biomass to issue a forecast. It's also not just a phosphorus problem. We also know that nitrogen has an extremely important role. Nitrogen has a secondary role to phosphorus in bloom biomass, but nitrogen has a primary role in bloom toxicity. We can do a lot of experiments in lab where we grab lake water and we add phosphorus, add nitrogen, or add phosphorus and nitrogen together. And we often see that phosphorus and nitrogen together stimulates blooms the most. This image here is from a September bloom a couple years ago, where it was nitrogen limited. But when we do these experiments, May through July, the phosphorus treatment really takes off, where the nitrogen treatment stays the same. But nitrogen is important later in the summer, and we always get the biggest increase when we do phosphorus and nitrogen together. A little bit more on nitrogen. This plot here shows nitrate concentrations near Stone Lab, and then the red triangles show the ratio of microcystins to cyanobacteria biomass. And as nitrate concentrations decline, we see a decrease in the toxins to biomass ratio. We see this decrease in nitrate concentration every year in Lake Erie and in Maumee Bay near Toledo and further east. We always see this decline where we get extremely low concentrations of nitrate in August and September. This graph here shows a plot of toxic strains. So I should have mentioned earlier that not every cyanobacteria cell can produce toxins. There's toxic strains and non-toxic strains. Early bloom, when there's a lot of nitrogen available, over 80% of the cells in Lake Erie have the potential to produce toxins. As nitrate declines, we end up with more non-toxic strains, and which is matched here. We see more toxins per cell here, less toxins per cell late season. And we can also do those similar bottle experiments where we spike in phosphorus and different forms of nitrogen and incubate them at different light levels. And then what we see increased microcystin concentrations when nitrogen is added, but also at high light levels. So nitrogen additions at high light trigger microcystin production. So it's not just phosphorus, it's not just nitrogen, but light also has an impact. So water clarity, cloud cover, examples for light levels. So what is being done to stop blooms? We talked a lot about phosphorus and nitrogen. So the goal for Lake Erie is to keep phosphorus and nitrogen on the fields during wet springs. We know it's going to rain. We know farmers need to fertilize. So the goal is to keep those fertilizers on the fields when it's going to rain. Again, like I said, I'm not a soil scientist, so this is coming secondhand information here. But placement of fertilizers near the soils, not just spraying the fertilizer on the surface. In Ohio, we are not allowed to fertilize on frozen ground. We're not allowed, this is inorganic fertilizers or manure. We're not allowed to spread those on frozen ground, and we're not allowed to apply them right before a rainstorm is in the forecast. This bill was passed and signed a couple years ago, banning fertilizer placement on frozen ground. A lot is being done looking at soil phosphorus test. So how much phosphorus varies from field to field and even within a field. So field A might need more phosphorus than field B. A lot of work is being done looking at better use management practices. So buffer strips, cover crops, fertilizer placement, two-stage ditches. What's difficult is that it's not a one size fits all approach. If every farmer went to cover crops, that might not solve it. If every farmer did buffer strips, that might not solve it. So a lot of factors need to be taken into concern, such as the soil type, the slope of the soil, what do the crops need, the organic content of the soils. So there's a lot that need to go into it. And I think that's the harder thing to do, is that there's no silver bullet. Farmers are already taking action. A survey that was done within the Maumee River watershed, 70% of the farmers agreed that fertilizer lost from their fields have negative impacts downstreams. And 70% of them are willing to do at least one more thing to help keep fertilizer on their field. This is a recent survey, I think about 5 years old now. But I believe if this survey was done 20 years ago, there would have been a much different response. When there's lots of rain and storm in the spring, farmers' fields flood, they turn brown, and then the corresponding ditches, streams, and rivers turn brown too. And then you see it out in the lake turns brown, then eventually turns green. So a lot of action is already being done. We can't be blaming farmers. We got to work with the farmers and give them the right tools and incentives to help them keep fertilizers on their field, because it's really a lose-lose situation. They're buying fertilizers, and when it rains, heavy rains, it just gets washed downstream and has negative impacts in the lake. I talked about wetlands in Ohio. 2 years ago, Governor DeWine in Ohio initiated the H2 Ohio program, which has several things that it's doing. But one of the things is that it's helping recreate wetlands. All the stars you see on this map are wetlands that are being reconstructed or converted from other lands into wetlands to help capture phosphorus in the stream before it gets to the lake. I think now this map shows 30 projects. They have increased it to like 75 or 80 within the past 2 years. So a lot of money is being spent on recreating wetlands to help reduce phosphorus. Ohio also has this harmful algal bloom research initiative, which is a collaboration of several state agencies. So every 2 years, the Ohio Department of Higher Education asks Ohio EPA, the Department of Ag, the Department of Health, and the Division of Natural Resources, what are the most pressing issues facing harmful algal bloom in your area? And then the state funds research projects to Ohio universities that are designed to provide real near-term solutions for a whole suite of issues. So some examples of the harmful algal bloom research initiative. One area of projects is tracking blooms from the source. So monitoring the tributaries for the nutrients and understanding, developing early warning systems within the lake that can help aid water treatment decisions. So tracking the blooms from fertilizers, phosphorus, and nitrogen in the river into the lake, and developing early warning systems. Another group of projects is looking at producing safe drinking water. So the timing of intake. Do you want to draw your water in at day or at night? Align it with wind. Do you want to draw water in when there's an east wind or a west wind? Once water gets to the treatment plant, how do you optimize treatment to remove microcystins? Before this project and before the Toledo water crisis in 2014, there was no set of recommendations on how to remove microcystins from water. Every plant did it differently. Now there's a recipe of looking at activated carbon and ozone treatments, to specifically remove microcystins from drinking water. Okay, let's say, hypothetically, microcystins get out of the treatment and into the distribution system. What happens? Do they just go straight through it into homes? Do microcystins bind to pipes? Do they accumulate in pipes? We don't really know. And then point of use projects. So, can your your under sink filter remove microcystins, or even Brita pitchers, do they remove microcystins? So point of use. And then projects looking at public health, identifying pathways to exposure. So drinking water is the common or it's the obvious route of exposure, but are microcystins in walleye or yellow perch that we eat? Can the microcystins be aerosolized? So if you're walking next to a lake that has a bloom, can you get exposure that way? Researchers at the University of Toledo are developing a blood test to determine have you been poisoned by microcystins or not. So it's largely a truly collaborative event. There's US agencies involved, nonprofit, and which the research projects are really driven by these 4 Ohio agencies. And then this is just an example of some of the universities within Ohio that are funded. To finish up, I'm going to talk about some research that my lab has been working on. We are developing a harmful algal bloom toxin forecast. So that forecast that I mentioned a couple of minutes ago that related phosphorus load to bloom biomass does not account for toxins. So we are working on a NOAA funded project that is looking at developing a short-term toxin forecast. And what we found, this plot here is kind of difficult to understand, but blue and dark blue means the model was correct. Grays mean the model was incorrect. And this top model output shows that when we run the models with microcystin production rates and water currents, we get a much better result than if we only look at water currents. So in short, biology matters. And for this site right here, which is called WE12, we were accurate about 80%, 85% of the time, asking will microcystis increase or decrease over the span of 7 days. So just that information alone would be really helpful to a water treatment plant operator. Just knowing if microcystins will increase or decrease over the week, so then they can prepare to treat that water. We are looking at new technology to detect microcystins. So the current method takes at least 8 hours from water sample to data. We are working with a company based in Boulder, Colorado, to develop a new test that only takes 20 minutes. So we're, is this new method, is it as good as the current method? So that's ongoing. We are looking at the effectiveness of algaecides in small lakes. So if you have a bloom going, can you put something in that lake to kill the bloom? There's copper sulfates and peroxides and ozones. So we're doing beaker-sized experiments, but also in a small privately owned lake that the Lake Management Society has asked us and allowed us to sample their lake pre and post ozone treatment. However, I'm highly skeptical that anything, basically anything that's larger than a couple acres, I don't think treating a lake is the way to go. We really want to prevent blooms. It's an issue of scale. So the amount of stuff, copper or peroxide, you need to put in to kill a lake or kill a bloom is a lot. And when you scale it up, you're talking about tons of copper. It's not feasible, and there's can be a lot of negative side effects. And we're also looking at saxitoxins, which is our different type of toxins that mainly occur in inland lakes. A lot of focus in Lake Erie and many other inland lakes has been microcystins. We know a lot about microcystins and what triggers microcystin production. We're developing models and forecasts, but saxitoxins are also common and they're also highly toxic. But we know very little what causes saxitoxin production. We also host dozens of researchers from all around the world to study harmful algal blooms and other Lake Erie issues. This is just a graph showing how many visiting researchers and projects we've had. We really bumped up our research activities at Stone Lab over the past 10 years. COVID took a big hit to our research activities, obviously, but we bounced back this year. And we're also constructing a new mesocosm facility right outside Lake Erie, so we can do larger scale experiments. We could look at large scales like this. We can put fish in a tank with microcystins to determine, are those microcystins getting into the fish, and then if so, does that pose a risk to anglers who like to eat the fish? So with that, I'll be happy to take any questions. And thanks again for inviting me to speak.
Thank you so much for that talk. We did have some questions come in. So the first one is, are dissolved reactive phosphorus and orthophosphate synonymous?
So that's always a good question. We define dissolved reactive phosphorus as the phosphorus that goes through a filter and is reactive with the chemical that you use to test for phosphorus. It's supposed to represent orthophosphate, but there are some nuances that it may not be exactly the same, but we assume, maybe not assume, but there's that caveat that it's supposed to indicate orthophosphate. I'll just say it. It's supposed to be the same, but it's not, but it's pretty close.
The next question, so with the decrease in nitrate, and the decrease in cells and microcystin, could that be due to a decrease in temperature in September?
So actually, Lake Erie peaks in temperature early September, late August, early September, and then starts to drop. There is research looking at the effect of temperature. If you hold nitrogen and phosphorus the same and you decrease temperature, some recent research has actually shown that will stimulate toxin per cell. So cool water may stimulate toxin production. That is still being debated, but the effect of nitrogen appears to be more stronger than temperature.
And then the next one is, does the N&P ratio, in addition to total nitrogen supply, also affect microcystin toxin production?
That's always a good question. The N-to-P ratio has a larger impact on which type of cyanobacteria will be around. Mainly looking at nitrogen fixing cyanobacteria versus non-nitrogen fixing cyanobacteria. Microcystis, the main one in Lake Erie, is a non-nitrogen fixer, so it has to have nitrate or ammonia in the water. Lake Erie has extremely high nitrogen values, extremely high N to P ratios. When the blooms first occur in July and early August, the N to P ratios are well over 100. As nitrate gets denitrified, that N to P ratio does drop, but and then the dissolved N to P ratio would drop, but the total N to P ratio stays typically above what we would consider limiting if you just look at that traditional N to P ratio. So there is an impact of the N to P ratio, but I believe the concentrations are important, but also the turnover rate of ammonia. What is really sustaining these sustaining microcystis blooms is ammonia regeneration. You can't measure ammonia in the water column because it's sucked up as fast as bacteria create it. So when that generation rate is high enough to support microcystis, the N to P concentration ratio really doesn't matter all that much. Because the generation is so high, microcystin is extremely competitive for ammonia. But eventually, even that runs out, and then we can see a shift to shift to nitrogen fixers. But for the most part, it stays microcystis throughout the year.
We have another question on reverse osmosis. So does the reverse osmosis water purification process remove microcystin?
I believe it does. I think there was the filters, again, this is coming from secondhand, but the filters can become leaky with chlorine, I believe. So it might depend on how chlorinated your water is. So there's something there. If you put microcystin in pure DI, run it through osmosis, reverse osmosis, it removes it. But when you start adding in other things to that water, the filter starts to break down, I believe. Not 100% sure, but I know that research is ongoing.
And it looks like the last question I have in is actually a multi-part question. So I'll read it all out, but we can break it down if needed. So can you please elaborate on the nitrogen toxicity connection? Is it strictly related to species composition? And then also, it seems like you have Delicospermum, which is an N-fixing. Are you saying that you see a shift from microcystis to those other species?
Yeah, that's so the first part about nitrogen and microcystin, the toxin microcystin, depending on congener, is 10 to 20% nitrogen by mass. So the toxin itself is highly rich in nitrogen. So it has to have ambient nitrogen to create this toxin, extremely high nitrogen rich compound. So that is a stoichiometry thing there. When there's less nitrogen available, they're not going to, microcystins are not needed for regular cell growth. It's a secondary metabolite. Why they're producing microcystins, we don't quite know yet. But when there's low nitrogen available, they're not going to produce, they're not going to waste that nitrogen, I guess one way to put it. So they need, they will use, they'll shunt resources towards growth when they become nitrogen limited rather than toxin production. And then the second part, what was the second part again?
The second part, so with species composition.
Okay, right,
it looks
right.
like
So
you have
microcystis versus Delicospermum. We do see Delicospermum in the Western Basin, not every year, but it's normally September, October. So after nitrogen gets depleted, microcystis sucked up all the nitrogen. Nitrates being denitrified, and then eventually we can get extreme nitrogen limiting conditions, which allow for the end fixers. However, I mentioned Dolichospermum in the central basin. That is very interesting because that Dolichospermum in the central basin occurs early summer when there's very little phosphorus in the water and extremely high nitrate concentrations. So the central basin Dolichospermum do not follow that general tenet of low N to P favoring nitrogen fixers. Iron might be limiting out in the central basin, so that nitrogen out there might not be available for growth, which could then favor a nitrogen fixer. But yeah, that central basin bloom is kind of unique. I've been looking in it for a couple of years now. It's hard to explain, really.
Well, I am not seeing any more questions, and we are at 4 o'clock. So I think we'll wrap things up now. Thank you again for your talk. It was great hearing about what's going on in Western Basin of Lake Erie.
Right. Thank you, and have a good rest of your seminar series.
Stop recording.
All right, take care, everybody.